Hey guys, so today we are going to talk about this portion is the sex genetics. So let me share the screen. It's two. All right, share. All right. Okay. Okay. Good. Let's get started. So this one. So earlier we talked about the different Mendelian genetics. We talk about different phenotypic traits, and we always mention about a word that is the autosomal. So whether that's autosomal dominant or autosomal recessive, those are all autosomal traits. Autosomal traits affect both sex, both male and female, and those are the 1 to 22 chromosomes in our body. And with this topic, we're going to talk about the sex chromosomes. So then we will kind of complete all these 23 pairs of chromosomes in our body. All right. So the sex chromosomes determine our sex. In male has the chromosome XY, female has the chromosome XX. So male is a heterogamic chromosome because it's different XY. Female is homogamic sex. That is the XX. This X and Y have different lengths. X is much larger than the Y, and Y only has 231 proteins encoded genes. X has more than 1,500 genes. So if we get a chance to look at the size, we can see that X is much bigger than the Y. There was a debate that maybe 100,000 years later, there will be no male because the Y is getting shorter and shorter. In the end, it's gonna disappear. But that's just like idea. Who knows what's gonna happen a couple 100,000 years later, right? Or million years later, right? So the thing, but the fact is that Y is much shorter, and there is some evidence showing that Y is actually gradually getting shorter over time. So there are several mystery over there in the Y chromosome. All right, so here is the sex chromosome XX and XY. We get this from our parents, apparently. So mother, father, both has their gamete. Mother have the oocyte, father has the sperm. So here you can see that can you see the, not so sure if you can see the pointer. Pointer option, okay. So mother if the gamete is either X or X. So it could be this X or that X, but either way, it's gonna be an X. And the father will give a sperm that is either X or Y. So it's either contain X or contain Y. If that X mate with this X or this X, the offspring gonna have two X, and the offspring gonna be a female. If the sperm contains this Y instead of this X, then Y mate with X, either this X or that X, the offspring gonna have one X and one Y. That will make this infant a male. So male is XY, and the chance is 50-50, right? Because mother can have this X or that X, doesn't really matter. But if the egg meet either this sperm or this sperm, it could be 50% male, 50% female, or 50% male. So that's the production of the offspring from the parents. All right. So X, both male and female has X, but only the male has Y. So let's look at the Y chromosome, and this is a Y chromosome. Like every other chromosome, it all contains this portion as a centromere. This portion is a short arm, and this portion is a long arm. So these are very typical to every other chromosome. Every chromosome has a centromere, short arm, and a long arm. When we do the staining, we can stain something darker with something shorter, lighter. When we stain, so here is, so we can see the segment, like the euchromatin is a portion that's not stand strongly. This portion represents the protein encoded genes. In contrast, we can have a portion that's stained with the histology staining that stand strongly is called the heterochromatin, and that represent the region is not very active for the transcription, so it doesn't provide the protein encoded gene. So with this stand strong area, a lot of these coding are not protein encoded genes. Okay. We actually in our chromosome we actually have a lot of these non-encoding genes. I remember only 2% of the genes are protein encoded. The other 98% of the genes are not. So that's the separation of the euchromatin and the heterochromatin. So if we have a quiz, this is gonna be the quiz question: that which chromatin contains stronger staining, which chromatin contains the protein encoded genes, and those protein encoded genes are involved in transcription because those genes will be transcribed to RNA and to produce the protein. So these portions are all common across different chromosomes. Short arm, long arm, euchromatin. There are some genes. It's for the protein encoding. there are some regions that's not for protein coding, heterochromatin. So those are centromeres. These are all very common across different chromosomes. Something unique here is D3. Unique here in the Y chromosome is D3. So in Y chromosome, we have these two ends. It's called pseudo-autosomal region, PAR. Means that this region represents some portion in the autosome. So those 1 to 22 chromosomes. And this portion is getting more interesting. Here, this portion is called male specific region in Y chromosome, MSY. So this region is, so these two are for autosomal, like not sex related. And this region is sex related, male specific region in the Y chromosome. And among these, the most important one is this portion. When we have the italic writing, it means a gene, a sequence of gene. So this gene is called SRY, the sex determining region of the Y. So this one is the key. You got the Y chromosome. If you don't have SRY, it's not going to be determined as a male. Only if the SRY is activated, then this person will develop to a male or sex organ structure. So that's the this again terminology, PAR, pseudo-autosomal region, MSY, male specific region in Y. SRY is the sex determined region of the Y. So how does this develop into male versus female? It must be coming during the infant development, right? Because in the very beginning, it's only zygote. There's no sex characteristics yet. Now one cell divide to 2, 2 to 4, 4 to 16, right? So it gradually expand. And at some point, this will develop into the embryo, right? And then embryo will develop into the infant. So the time to develop into the infant is about 8-9 weeks after the fertilization. And so just right before the 8-9 weeks, the choice of developing either become a male versus a female is about the 6 weeks. So before 6 weeks, say 5 weeks, we can the sex structure is not shown yet. It's not clear yet. But by the 6 weeks, SRY become important. SRY will develop the male sex organ, like penis, right? If there's no SRY, then the infant will by default develop into the female structure. So let's see how it does. How does it work? How does it develop into sex differentiation? So by the 5th weeks, so that's halfway through the embryo development, we have this the body has this organ. This organ contains the gonads. We have 2 gonads. We also have 2 tracts. One is called the Wolffian tract, The other one is called the Mullerian tract duct, Wolffian duct, and the Mullerian duct. So these two. And so that this is unspecified yet. We have the gonads. These gonads could become either an oocyte, the ovaries, or the testes. So if it's the ovaries, then this will become the uterine tubule. If this is the testis, it will fall down here, and these will become the vas deferens, the male sex tract, right? So that's that. So by the 5th week, the embryo has developed 2 unspecified gonads. These basically 2 unspecified gonads, as well as these 2 tracts. If we have the SRY, then Wolffian duct will develop into male. If we don't have the SRY, the Mullerian duct will develop into female sex structure. So say we have the SRY, then SRY will turn this unspecified organ to cause the degeneration of the Mullerian. This Mullerian will degenerate it, and the Wolffian duct will develop into the ductus deferens or vas deferens. Gonads will fall down here to become the testes, and also penis will be developed by about 8-9 weeks. We will see, using the ultrasound, we can identify this baby is a male, right? It's a boy. That's the activation, that's the result of the activation of the SRY gene. SRY gene is the key, okay? The SRY gene, this gene will be transcribed to several factors, and this transcriptive factor will then affect other genes. Altogether, it will start to synchronize everything to move these development into the male sex organ development. If the SRY is absent, is either there's no Y chromosome, like the typical female, it has the XX chromosome, it doesn't have the Y chromosome, there's no Y, there's no SRY, or the SRY is silent. There are some disorder that the SRY is mutated, or SRY is disappeared, it's missing, and then this body will develop into the female. So the Mullerian duct will remain, and gradually there's no SRY, then the Wolffian will have no support to sustain its availability. It will degenerate it. Then gonads will develop into the ovaries. This Mullerian will develop into the uterine tubule. And so we will have the female sex structure, including the ovary, uterine tubule, uterus, vagina. So that's become the female. So by about 9 weeks, if there's no SRY, there's no penis, this baby, through ultrasound, we can see that this is it's a girl, baby girl. Okay, that's that. So the timing of the sex development is about 5 to 9 weeks after the fertilization. And with the SRY, the Wolffian ducts will develop into the male sex structure. Without SRY, the Mullerian ducts will develop into the female sex structure. So the default is if there's no SRY, it's gonna be female. Then the question is, how does the SRY work? We know that SRY will cause the male. Then but here we want to see how does it cause that? There are several things we need to have to form to form a male. Which include you need to have three things. One is that you need to kind of suppress the Mullerian ducts, because Mullerian ducts is the default. Both male and the female has X, and the default development from the embryo is to form the female. He has the potential to form the female. If there is no interference, it will form the female. So we need to first suppress the development of the Mullerian duct. Second is that we need to develop two things. One is the internal organ of the male, such as the epididymitis, ductus deferens, vas deferens, seminal vesicles, ejaculatory ducts. So these are internal. We also need to develop the external feature, the penis and the scrotum, for example. So there are three major things that SRY need to achieve. And how does SRY achieve that? So SRY will first secrete the stimulate the cells called Sertoli cells to secrete the hormone called anti-Mullerian hormone. So that will suppress the development of the Mullerian and cause it to degenerate. Remember the Sertoli? We will have a picture. Sertoli is the cells in the matured body is used to neutralize the sperms. This is also called the sustentacular cell, Sertoli cell, okay? And then we also need to have internal. The internal is the Leydig. This Leydig cell is also called interstitial cell. We mentioned about this one earlier. Interstitial cell, Leydig cell. And these Leydig cells are interstitial cells. They are also in the testis. These cells to stimulate these cells to secrete sex hormone. The male sex hormone is testosterone. So with this testosterone, the baby can start to develop the male sex tract, including the epididymitis, ductus deferens, or this also called vas deferens, seminal vesicles, and ejaculatory ducts. So one thing you need to know that by about 9 weeks of time, or 6, 7, 8, 9 weeks of the gestational age, the baby boy already secretes high amount of the testosterone in the mother's body. Okay, yeah. All right, so that's the develop the internal sex tract. Now external, we need a different pathway. That is the DHT. DHT can promote the development of the urethra. This is the duct inside of the penis, prostate glands. Although this is kind of like internal, but this is stimulated by the DHT, penis, and the scrotum. So where do we get the DHT? We get the DHT is dihydrotestosterone. So this is converted from the testosterone. Testosterone is converted to the DHT, and the testosterone is secreted from the Leydig cells. Okay, so this conversion is also very important. If this conversion failed, if we don't have enough enzyme, if we don't have the correct enzyme to convert it, then the baby would only develop the internal portion, but not the external features. Okay, so this table contains a lot of important information you need to know. That which cell secretes what? You know, that's a quick question. That where is Mullerian anti-Mullerian hormone is secreted? It's from the Sertoli cells. Where do we get the testosterone from? The Leydig cells. Where what's important to develop the external sex feature like penis, scrotum, etc. We need DHT. Okay, so that's that. All right, so that's the here, just showing you the picture, give you kind of like a visual memory, refresh your memory about this important cell in the testis. We have the Leydig cell. Here is the interstitial portion. Leydig cell, they secrete the testosterone all the time. It's not just the infant, but throughout our body's development, we have these Leydig cells to secrete the testosterone. And that builds in the baby, it helps to build the external like penis, scrotum, etc. When the boy start to develop, it helps to build muscle, bones, hair, etc. So that's the Leydig and the Sertoli during the embryo. This is used to secrete the anti-Mullerian hormone to suppress the development of the Mullerian duct. But after the 9 weeks, we don't need them to secrete anti-Mullerian hormone because Mullerian is already degenerated. Their next job is to neutralize the sperm. So the Sertoli cells is right here. Their function is to neutralize this sperm to promote the sperm development. So that's that. Also, last time we also mentioned about the smooth muscle. So this is for the ejection that squeeze the semen, not semen yet, but fluid here from the tubule into to eject it out, right? So that's that. And then we can develop into this male reproductive system. We will have the testes or testicles. This is the vas deferens. So we can get the idea that it's coming from the, this is the gonads. It's similar like females, ovaries. Vas deferens is similar like uterine tubule. And then we have the semivesicles. Here is on the back of the bladder to provide the 75% of the fluid in the semen. And here is the prostate gland. And then here is the urethra, here is the penis. And the scrotum containing the testicles. Urethra is the ducts of the urine system, the pee coming from the kidney. And then here bladder is a place to store the urine. So that's that. And then we know about this sex development. Let's take a little bit detour to look at what if things not work as expected. So we call this group of the condition is a DSD. It's called disorders of sex development. So this could be caused by so many things. As I mentioned, before, SRY is very important. Y chromosome, even though through the chromosome staining examination is called the karyotype, we can know that we have the XY and we thought that you got a baby boy. But if the SRY is missing, then we will not develop into the male. Or throughout the SRY process, the development of the testosterone is important. The secretion of DHT is also very important. So any steps, if there's any missing part, we will have the develop into the condition called the DSD, the disorders of sex development. This is also generally called the intersex study. Sex organ is ambiguous. That's not very clearly immediately recognizable. This used to be called, a very long term is called hermaphroditism. And this hermaphroditism is coming from the Greek word herms and Aphrodite. And so this is like this is like Greek gods, right? Like, and their son is, you can look it up, you know, that's the story that their son has both male and a female like combination. So that's this term coming from. So let's look into what's the common DSD that in the clinical that we may see. So here is some like karyotype. This is the first step to recognize, to examine the if there's any concern about the sex development disorders. So here I show you the, so when we scan the chromosome, we can present, and we have 23 pairs of the chromosome from any cells. You can swap the skin cells in your mouth, and then you can just look into the nucleus, and then we can stain this. You can see that yes, a dark band, light band. We sequence this, we line this based on the size from the longest one to the shortest one. And in the end, we have the XX or XY. So that's right here. So this is typical normal karyotype. When we present this under my microscope, this is the karyotype. This is also terminology that you need to memorize it. Karyotype is a way to present all 23 pairs of chromosome all together. And basically we can get something like if we have missing part, if we have extra part, right? So normally, healthy situation, we have typical 23, okay? In some situation, we may have missing part, like this one. This one is common. This one is one of the common disorder that we only have one X. We don't have the other. We don't have Y. We don't have two X. So here you can see that right here. This one is called Turner syndrome. It's also called 45X. It means that typically we have 46, 23 times 2, right? Here we have 45. And the 45, the missing part, the disorder part is X. So we only put 45X. We can also have another two types. So this one affects female because it doesn't have the Y, so it doesn't have the SRY. So this will by default develop into a female baby body. However, it only has one X. So the proteins, the gene expression from single X is not as strong as from two X. So there will be some features not fully developed. And so that affects the female. The female doesn't develop into the full like female structure. We also, this is more common, but the less common is these two. We may have double X. So we will have one egg with two Y, one X with XY, and the other sperm from the father carry Y. Sorry, yeah. Anyway, so we have the double Y, basically double Y. So this one we call it 47 because this one it's not just 46. We have one extra, so it's 47, and the disorder portion is the XYY. So here is XYY. It's 47 common XYY. So that's the typical way we label it. And this one affects what? This one affects male because we have the Y. We do have the Y to produce the SRY, to produce the male structure, but we have too many Y, and this will lead to some disorders. Another typical one is this one. It's called double XY. So this one is 47 XXY. This one also affects a male that we have the Y, but we have too many X. So that affects it. It's not just the chromosome structure disorder, they affect the phenotype. So these are commonly found in the chromosome. We also have some disorder that is 46XY or XX. Let's take a look. So if in the karyotype, it may appear very normal, but the development is not. So this is called the 46XX. So it's very typical, 46XX. If you see this 46XX, you will think that this is a girl, right? So this is a female. But turns out it developed into male, okay? So this is called XX male syndrome. It has a lot of X, so it has a lot of potential to develop into female. However, so this is a typical XX female. However, earlier when we talk about this, meiosis, right? We know that the chromosome can have a crossing over. Some part will switch to the other. And this is the crossing over, result of the crossing over that Y chromosome cross over their SRY gene into X. So in this situation, the karyotype show you double X, but it's a male. It has the SRY gene and it promotes the development of the male structure. So this is called XX male syndrome. We can also have the XY. XY is supposed to be a male, but so X46 XY is supposed to be a male, but it's developed into a female. The reason is that its SRY gene is either mutated or missing in the Y chromosome. So we don't have the SRY gene, we cannot develop into the male structure. XY female can have two different categories. One is the SRY gene, like this one here. The other one is that even though we have the SRY, but we have other hormones deficiency. So we still have the XY, like supposed to be a male, and the other hormone can, as I mentioned before, throughout the entire process, we need sufficient we need the sufficient testosterone and the DHT to develop internal and external male sex structure. So even though we may have a good beginning, a good SRY, if any step goes wrong, the development into male may not be completed. So that will be considered as another, like a subtype of the XY female syndrome. So let's look at what could be that. So this is the various 46. So it has the typical standard number of the chromosome. It has the XY, so it should be a male, but it developed into the sex development disorders. So here is the pathway throughout the infant embryo development to develop into these sex structures. And so let's not worry about the green first. Green is a point where there is a mutation that the gene doesn't work, doesn't function. So let's look at the normal first. We have the SRY, SRY turns on, the X on the testis to convert this Leydig cell, the interstitial cells, to produce what? To produce testosterone. So it will convert the cholesterol to produce testosterone. Testosterone, as I mentioned earlier, that is the steroid hormone. The steroid hormone are the derivatives of the cholesterol. So, and then testosterone is important to develop internal sex structure as well as to convert to the DHT to develop the external sex structure. So testosterone can develop into the internal epididymitis, ductus deferens, seminal vesicles, ejaculatory duct, and converted to the DHT. We need an enzyme, and to convert to DHT acts on the external portion to develop urethra, prostate gland, penis, and the scrotum. It also stimulated the Sertoli cells to secrete the anti-Mullerian hormone to suppress the development, to cause the Mullerian ducts to be degenerated. So that's the normal process, right? Now, a lot of things can go wrong here because it's not like a single step. It has many steps, especially during the embryo development, some of the enzyme probably is not secreted normally. So the very first one is the SRY. So let's look at this point of mutation that caused the problem. So one problem is the SRY. So we may have the XY, but the Y doesn't carry the SRY, or the SRY is mutated. So it won't turn on. If this one doesn't turn on, if we don't have the SRY, this XY male will not develop into a male. It will develop into female. It will be fail in the very first, in the beginning. Okay, so that's the common one that we see. Another common one is that the patient may not be able to have the capacity to convert the cholesterol into the testosterone, or doesn't develop into enough the testosterone receptor, because this testosterone need to act on the cell to promote their differentiation into different organ. If the receptor is disappeared, then even though we have a lot of testosterone, we have no receptor, this organ won't be developed. We also need to convert the testosterone into DHT. This conversion requires an enzyme called 5-alpha reductase. 5-alpha reductase may be deficient. If it's deficient, then the patient will be able to develop something internal, but not external. So it may still generate sperm, will be able to, with the testosterone, to build the body, like hair, like muscle, look like a man. But without DHT, it will not develop the penis external organ. Or the patient may not have, may have a lack of the DHT receptor. Then this patient will also develop into the, may not be able to develop the external sex organ. So there are several possibilities that we may have the XY female syndrome, okay? So among all this, I want to use this as an example because this is more like a common one, that babies was first born recognized as a female because they don't develop the external. However, they have everything internal. And they, in the beginning, they may be treated as a lady, as a girl, baby girl, and raised up as a girl, but may act like a boy. And later puberty that generated the hair, and then realize that they actually are boys. So this is something that we can take a look. So of a reductase deficiency, it cannot convert the testosterone into the DHT. During the fetus, there will be no external because DHT is needed to develop the external portion. So everything will be ready, but not the external. So by doing the ultrasound to check it, we may see it as a girl, and we raise the baby as a girl. But during the puberty, a lot of testosterone got secreted and released from the gonads. These gonads stay inside of the body, is not developed into the scrotum, and a lot of male feature will be visible. And in the same time, the female's feature won't be visible because this person has no problem to suppress here. has no problem to suppress the Mullerian duct, has no problem to, and there's no ovaries, there's no uterus. Instead, they do have the testosterone and sperm to be developed. So they don't generate breast like a female. Instead, they have everything like a male, except that external structure. So this can be fixed by some surgery to fix it and to recognize them as a male. If its female doesn't have the 5-alpha reductase, then that's okay. It will look like female. But if it's a male, then this 5-alpha reductase is an issue. Okay, deficiency is an issue. So that's that. And as I mentioned before, another group of the sex disorder of sex development is the chromosome either missing or extra. This missing or extra is called the Aneuploidy. Aneuploidy is the group of the condition that we have either missing or have an extra number of the chromosome. And here we are talking about the sex chromosome. So we can have the XXY or XYY, right? And if this can also be happening to the autosomal chromosomes. So for example, if we have one extra chromosome in the 21 chromosomes, we may have the patient to develop into the Down syndrome. Okay, so this is common one. So this aneuploidy is the term to describe the number of the chromosome is not normal. So it could be missing or extra one. Related terminology is the euploidy that refers to the normal set. Everything is normal. Polyploidy is the one that we have not just one more. We have the whole set. We have not just 47 or 45. We have 23 times 3, something like that. So the extra set of the chromosome. So just the terminology. And this could be the quiz question, right? Which one referred to that we have missing one chromosome, or we have one extra chromosome? Was the one to refer to the normal set of the chromosome? So with that aneuploidy, we may have some condition that is the missing one. Missing one is called monosomy. The monosomy X means that the 23rd chromosome should have either XX or XY, but it's only X, so it's monosomy X. This is called the Turner syndrome. This affects the female. We may have extra chromosome, which is called the trisomy. Trisomy 21 is the one to cause the Down syndrome. That one extra 21 have higher chance to develop so many different other diseases. The reason, the cause of the aneuploidy coming from the cell division. The reason that we have one extra chromosome or missing one chromosome is because that during the division, the sister chromatids supposed to be divided, but they are not. They are not disjunction. So this disjunction is not disjunction. So it's called a non-disjunction. And that's cause that sister chromatin is supposed to be disjunctioned to two different daughter cells, but they are not. They basically fall into one. And then in that egg or that sperm, we have two chromosomes there. And another egg or sperm, we miss one chromosome. So that's that. So here is some example that we may lead to the extra or missing one chromosome. So for example, it may be caused from the first meiosis. So here is the first meiosis. This is second meiosis. So it may happen from the first meiosis, or it may happen in the second meiosis. That in the end, we supposed to separate the sister chromatids, but turns out they all come into here. The other one is none. And then we will have missing, basically. This is zygote, so we may get the pink is from the other side. So this is sperm. Yeah, this is all sperm. We get the mother side, but not. We're missing one chromosome from the father. And here is a sperm. It has two, but plus mother, we have trisomic 3 chromosomes here. So that's the non-disjunction is a cause of the aneuploidy. And the aneuploidy in the sex chromosomes can lead to some disorders. There are 5 types of aneuploidies in disorders of sex development. The major one is the X0, 45X. This is also called Turner syndrome. This one affects female. There are some other one, the less common one, but also common, not as common, is that 47XYY or 47XXX. This 47XYY or 47XXY. This 47XXY is also called Klinefelter syndrome. And these two affects the male. They contain the Y, they have the SRY gene, but they have either too many Y or too many X. So that's that. So there are several type of aneuploidies to lead to the sex development disorders. So in addition to that, we may also have the body develop into some other sex orientation condition. For example, that the transgender refer to the person that's genotype and the phenotype are normal, but the identity, the way that he or she see him or herself is not consistent with the phenotype and the genotype. So that's a transgender. Another terminology is the transvestitism. This is a situation that the male like to wear female's clothes. So that's another condition that just you need to just know what this term refer to. Another common one that we been talking about it in our society is the homosexuality. This means that phenotype and the genotype are consistent, but the desire of the attraction toward the same sex, toward the members of the same sex is very strong. So that is the homosexuality. All these, condition that we don't really know what is the cause. However, this is not unique though. This is not unique. This is just we don't have the understanding yet. Why people will be physically or mentally attracted to the same-sex fellows is not known. It's not clear, but it's there. We just don't understand a lot of things. We don't understand our body. So this happens. We have found these kind of things in other animal species, as well as this happens throughout thousands of years of human history. So this is not unusual, and over time, we unfortunately have some tendency to blame this group of people, which is a shame. I feel it's a shame that for no reason, there's no reason that heterosexuality is better, is superior than the homosexuality, right? We don't know them, doesn't mean that they are wrong or they are worse, right? So a human being is a human being. To apply any pressure or strength on them is really a shame. It's really, yeah. So I was, you probably know that I was from Taiwan, and I'm very proud that my original country, I'm now in America, my original country was the first, was the first Asian country allow same-sex marriage. I think that's great achievement of human being in that society, because there's no reason to see them as a secondary citizens. They should and they deserve to be loved and also to love. Besides, they didn't do anything wrong. They probably will be more royal than a lot of our heterosexuality people. A lot of this homosexuality, they love each other, they support each other for decades. It's really a shame that see them as a secondary citizens. And we have the society to provide them some protection or the rights, the equal rights, which is, I think it's very, very, it should be done. It's not like something like advanced. This should has been done long time ago. Yeah. All right, so that's that. So here is a summary of the what we have learned. So here basically a lot of thing is the terminology, okay? So in the, this is the timing, fertilization. Once it's fertilized egg, zygote is formed, we already know that it's either XX or XY. So this is determined during the fertilization, the very first day. And then through the development, several things can happen, right? So gonadal sex start at about 6 weeks. Is Mullerian or Wolfian ducks will start to, one will degenerate, the other one will develop into the specified sex. So that's the gonad part. Then by about 8 weeks is also the time that the embryo will become an infant. These phenotypes, whether they develop the penis or not, basically, SRY to promote the development of the penis or not, will be appear. If there's no penis, through the ultrasound, we may see it as a female. If there's penis, we will see it as a male. So timing, so say you have a pregnant wife, you want to do do you want to know the sex? What's the timing to do it? About 9 weeks later, right? 9 weeks after the fertilization. 8 weeks after the fertilization. So that's the first time we would probably go check the ultrasound. And then gender identity, that the person identify him or herself as a male or female, start from the childhood, right? So and also sexual orientation, that he is or she is attracted to opposite sex person or the same-sex person, is also start very young age. Can you plan that? No. We see parents will know that they know their kids' orientation very young age. So I remember that when I was in like elementary school, we can see that, you probably see that too. Some boys will act like a girl, some girls will act like a boy. And for those girls act like boys, they probably be okay. But for those boys act like a girl, a lot of time they got bullied. And it's really not right. No, we should not do that. We need to protect them. They are just kids. And so personally, I know that sex education in younger age is debatable, but as a parent, I would teach my kids that we need to respect people that has different sexual orientation. And we are not better than anybody else. We have our own weakness. And we are just lucky to live in a society that we are more accepted than others. Doesn't mean that we are better than them. We are just luckier. All right. So another topic is called sex ratio. This sex ratio can be determined at different times. So sex ratio is the number of male versus female. This can be determined at the time of the conception. So that is the XX versus XY. So XY versus XX. And also the at birth with penis or not. And also at the maturation, that's the secondary sexual structure or characteristics be recognized. So that lead to three types of sex ratio. The primary sex ratio is a sex ratio at conception. Secondary sex ratio is the sex ratio at birth. And the tertiary sex ratio is the sex ratio at the maturity. And then let's talk about another topic. It's about the traits. Just like what we talked about in the autosomal, the autosomal phenotypic traits, right? The height, the hair, the curved hair or straight hair, right? So there are also some traits locate. These traits are expressed from the allele located on the sex chromosome. So traits inherent on a sex chromosome include the Y-linked traits. So this coming from the Y, so this is all about male. But this number is rare because Y-linked traits are fewer. The Y chromosome has fewer genes, so the traits linked to Y is rare. We also have X-linked traits. These X-linked traits are the traits caused by the allele located in X. And in contrast, if the traits coming from the autosomal chromosome, these are the autosomal traits. These are coming from controlled by the 22 pairs of the human autosomes, non-sex related. So that's our last, maybe couple weeks ago, the Mendelian genetics, we talk about the autosomal recession, autosomal dominant traits. That's the autosomal traits. That's same apply to. It doesn't really matter whether you are male or female. It's equally applied to both sex. And so these though, among all these, Y is only male. X though can be male and female, but X related to mother and son more. The reason is that X-linked traits. So in the daughter though, let's see the daughter. Daughter get X from one mother, one father. So a lot of time, even though one X is bad, okay, say this X cause some disease, something like that, she has another X to correct it. So say this X lack of some enzyme, causing this person lack of the enzyme to produce something. The other X can produce that, so it can compensate it. And this is also, earlier we talked about the dominant effects, right? So as long as you have one, you can cover it. So a lot of recessive traits, like lack of the enzyme or something, will not be shown in female. Female will not know that he carried that mutation. However, son will be different. The son will carry X from mother only, Y from the father. So this X becomes very critical. This X, if this is the healthy X, like a mother one, then the son will be okay. But if the mother carry a disease X, lack of the enzyme, etc., in the mother, she will not express any illness because she still carry a healthy X to compensate it. But if unfortunately this recessive traits is passed on to the son, then the son doesn't have another X to compensate it. This recessive X, this ill X, will become the problem, will cause the disease to the son. So that's the common condition that we see in the X-linked traits. Because if the mother has the disease, a lot of times they cannot have the baby, they die early, so that trait will quickly disappear. But if the mother carries the recessive traits, the mother will still, live long and have a baby. But they don't know that whether that recessive traits may pass on to the son. And so the son may disappear, may not be able to live long, and may cause several diseases. So that's that. So recessive allele from the mother can pass on to the son. And that's the major concern in the X-linked traits. This also happens to that if the mother has a grandfather, he or grandfather's brother has the disease, then this daughter may carry that recessive traits, and that recessive traits may skip one generation to the next generation. So the male inherits an X-like traits only from the mother. And so it really depends on whether this mother has a recessive allele or not. If she has, there's 50 chance that she will pass that recessive allele to the son. A female will receive 2 copies one from mother, one from father. So she doesn't really matter. If he has the recessive allele, she will know. He will know, so might not be able to pass, might not give the birth or something. But even so, she will definitely get a healthy X from the father. And as for from the mother, doesn't really matter whether that's healthy or not, because she already has a healthy one from the father to support her to live, right? So that's the X-linked traits. So here is an example of the X-linked recessive traits. This is a disease that causes the skin to develop a lot of what's that? Itchiness. So ichthyosis. That's the skin built up a lot of dead skin and make it very dry and also itchy. And so you can see the situation here. Here is the male, female. And if the grandfather has one, will pass on to the daughter, right? This is not like a fatal disease, right? So it may still give the birth to the mother, the daughters, okay, daughters. Now this daughter will not show it because this is a recessive. He got one good gene from the mother, and the one failed gene from the father. And since she has a good gene, good X here, it compensates the failed one. So she will not show any illness. She met a boy, they got married, they have a son. Then this son will carry that X here and the Y here. So he only has one X. And unfortunately, that X coming from the recessive allele from the mother. And he will show this illness too. So that will be a condition that skip one generation to the next generation. So that's that X-linked recessive. There are some traits, it's not X-linked, but it's sex-limited. For example, the sex limit means that only one sex, for example, the cow milk very low in saturated fat. This is it's a good thing, right? Because less saturated fat, we may have more unsaturated fat, and some unsaturated fat may provide the essential fatty acids. So that's a good thing. But this will only affect on the female, but not male. So this is sex-limited traits. We also had sex-influenced traits. That is that it influenced, not limited, but influenced by the sex. For example, the baldness. This can happen to male too, male and female, but it's influenced by testosterone. So with the present of testosterone, this condition can be worse. So this is sex influence. So we have sex limited. That is only happens to one type of the sex. It's because the condition is there. Sex influence is that it can happen to both sex, but the one sex has stronger effects than the other sex. All right, so that's that. Now, in the end, I want to talk about one topic that is also sex genetic related. That is about this little cat. So this is called calico cats. Calico cats are these cats that has these different random spots of the skin fur color, right? Very cute, right? People like it. I like it. I don't have the cats, but looking cute, right? So the question is that yes, is yes. Are calico cats always female? The answer is yes. Okay, then why? So this is about sex. This is sex genetic question. And we want to answer why, and we will apply this to human, okay? So this is something unique in female that is called X inactivation. So here we have female. Female has two X. Now, in the zygote, the first time we have the fertilized egg. That egg contains two eggs. That one egg will be divided to two. Each one will have two eggs. To 4 to 16, right? At some point, through that embryo development, each cell has two eggs. But the cell doesn't need two eggs, right? Because in the male, he only has one egg and it works fine. So the female will try to save energy and only use one egg. And the other eggs will be inactivated, okay? So say you have, now you have 8 eggs, 8 cells, right? 1 to 4, 4 to 16, say 16 eggs, 16 not eggs, 16 cells, this embryo. These 16 cells will start to inactivate in each cell. Will start to inactivate one X. This inactivation is random, so it may inactivate the one from the father, inactivate the one from the mother, inactivate the one from the father. So it could be random, and that's called the X inactivation. So that is what's going on. By the time an embryo consists of 8 cells, it's probably 16 because 1 to 2, 2 to 4, 4 to 16. There won't be 8 cells. So somewhere like that, that time. So that's actually very early. It's probably like less than 1 week after the fertilization. In each cell, one X will be inactivated. Okay, and that X to be inactivated is randomly chosen. So because the X, the person got one X from mother, one X from father. So which one will be inactivated is really random in each cell. Okay, all right. Once the part got inactivated, after division, the same pattern will be passed on. Those inactive ones will remain inactive, right? This inactive X will become a very unique portion. It's called a bar body. So if we look into the skin, like in male, we don't have it. In male, we only have one X. We have nothing to be inactivate because if we have only one X, right? And there's no random inactivation. But in the female, any skin, you examine that the cell, you will see that only one X and the other one become inactivated. And we see it as and it's called a bar body. This is first recognized from the Mr. Barr must be, right? Dr. Barr. And this is a technique that the Olympics is using to determine the gender or the sex of the athletes. We cannot just check if he has penis or not, right? It's just unreasonable to check that. So the way, more like a humane way, is to get the skin and to check if it contains the bar body, then we know that, okay, this is female. Right, so this is the diagram that I show you. Here we have the original egg, fertilized egg. It contains one maternal egg, paternal egg, mother and father. Green is mother, blue is father. One cell divided to two divided to four. About here, one will be degenerated, and that degeneration is randomly inactivated. That inactivation is randomly picked. So it could be the fathers, could be the mother, could be the father, could be the mother. So it's random, okay? But once it's chosen, then the following generation will have the same pattern. This group, say one embryo, sorry, one cell divide to so many, and this branch out will have the same pattern. This branch out will have the same pattern. If this start from the one that's the mother chromosome is inactivated, the following cells will have mother chromosome inactivated. Here, if the father chromosome is father X is inactivated, then following that, every cell has father X inactivated. And all our bodies start from the group of cell, divided, then that initial decision will make the following portion have the same pattern as the initial one. So when we develop into the skin, we may have all these coming from the same pattern. That is, say this one, that is the mother portion got inactivated, and this portion are the skin cells are from this cell that is the mother X is inactivated. So that is the X inactivation in female, and that leads to the firm color of the animal, because the color of orange is located in X. So this cat may have this X to express orange, this portion to express white. And in the embryo, he has both. He can express white and orange. But by the time the cell developed and the little cat is delivered, some portion of the skin will only express those showing orange color, but other portion of the skin will show the firm color of white. And that is quite random, but it will show us this region will be orange, that region will be white. And sometimes we have the another gene that is causing the darkness of that color presentation. And so we have different spots that has different color. So yes, calico cats are female. And the reason is that their firm color is determined by the allele located on the X chromosome. And that is either orange or non-orange. So all the calico cats are female. One exception is the male one. If the male is a disorder, contains the XXY disorders, it has one more X, then it may have both X. And this male, this is Y, so it's a male, may generate the pattern look like a female condition. So this is very similar to the condition in the XY female condition in human, that male will also express some female condition, right? Female feature. And so that is the female. Then your question is that what happens to the male? The male will not have this spot, will not have orange or white. It will be just one X, and that one X can be either orange or white. So the cat will develop to either the pure white cat or pure X, pure orange cat. It's not entirely orange. It has some like a darkness to it. So it will appear more like chocolate color. Another gene is called dilute gene. This dilute gene is not in the X, or it's not in the X. They will affect the darkness of the firm. So if it is orange, then it will be light orange or dark orange. If it's the calico, it will have the spots like lighter orange or darker orange. If it's the male, this male is called tortoise shell cats. And that male will have either pure white or pure orange or dark orange, like the chocolate color. So that's that. So that's the inactivation. So here, a little bit summarized here. In female, one important feature is the X inactivation, and that is observed, actually is discovered through the research on the calico cats. And the one to observe that, to discover it, is someone named Barr. So he later named this inactivated X chromosome to a term called Barr body. This happens during the early embryo development, very early, like almost like the first week of the development. And these effects of X inactivation can be noticeable, expressed in certain tissue, for example, in the cat. In the human, it's less noticeable is because we don't really have color form in S chromosome. We don't have that. Our skin color only D color, that D diluted or not, it's not a sex linked trait. It's an autosomal trait. And with our skin color actually is affected by many factors, not just one. And also, so the X may generate different hormones. That hormones or enzymes circulate the entire body. So even though it's only generated by a certain group of the cell, overall you don't really see that difference because it's all in the body. The amount is very much the same. So that's that. All right, then we finish this sex genetics, and let me know if you have any question. All right, let me see. How can I quit? Exit that. Stop sharing and stop recording.